Welcome everyone, and thanks for joining us for our webinar, Boosting Your Biologics Research with Chemically Intelligent Software. My name is Karim. I'm a Marketing Communications Specialist at ACDLabs, and I'll be your host today. We have two presenters joining us. The first is Dr. Eliot Randle, the Managing Partner and Co-founder of Digital Lab Consulting. He has over 25 years experience in the biopharmaceutical industry across the entire value chain from R&D to manufacturing and clinical, and he'll share some of that experience with us. Our second presenter is ACDLabs' own Dr. Kareem Kassa. He's the Senior Director of Customer Success, and he's worked with us for over 19 years. Before joining ACDLabs, Kareem spent 6 years as a medicinal chemist. I'm going to pass off to Eliot very soon, but first I want to provide a brief tour of the webinar interface for those who haven't used it before. On the right-hand side of your screen, you're going to see a panel. It might be collapsed into an error strip, but you can hit the button at the top with the arrow just to expand it out. You can use this to adjust sound settings and submit questions. Feel free to submit questions in the questions panel at any point. We'll take them up at the end of the presentation. If you're having issues with your video and audio that you don't know how to fix, you can close the webinar and re-enter. That should solve most problems, but if it still doesn't work, let us know in the chat and we'll try to help you out. With that out of the way, I'll pass things over to Eliot, who will start us off by talking about the field of biopharmaceuticals.
So yes, today I'm going to talk a little bit about how biopharmaceuticals are changing the future of medicine. So let's start with a few of the key facts about biologics and give an understanding of why they're growing in importance and interest in the market. So first of all, compared to small molecules, biologics tend to have a relatively low rate of attrition from preclinical to moving to the market. So less failures along the development route. Also, more generally in the market, you'll see an increasing proportion of the top 100 selling drugs are biologics, and that includes quite a lot of the top 10, including the top 2. So the top 2 being Humira from AbbVie and Keytruda from Merck. And since 2014, so going back about 6 or 7 years now, biologics drugs have actually accounted for more than 90% of the growth in net drug spending in the industry. So, these things together have led to a lot of interest, a lot of biotechs and pharmaceutical companies focusing on biologics and sort of riding that wave of interest in biologics in the market. So let's focus for the purpose of today on just one type of biologics, and that's antibody drug conjugates. So antibody drug conjugates, they leverage the specificity of monoclonal antibodies to target drug delivery, for example, to tumor cells, whilst they're minimizing damage to healthy tissues, so reducing side effects. So they're using that specificity of the antibody to deliver what we could call the payload or the drug to the site of or where it's needed most. So in this case, a lot of the ADC therapies are in cancer treatment. So targeting the payload, the drug payload, to the tumor site and letting it do its work where it's needed the most. ADCs can also overcome issues associated with traditional small molecule therapies, so things like clearance and cellular internalization as well. And going back to that earlier point about the market interest, specifically the ADC market is predicted to grow to over $13 billion within the next 5 years. So again, a large growth in that market is I think currently at about 2 or 3 billion. So fairly sizable to start with, but growing pretty fast. But one of the issues that we see with ADCs is they have a very complex molecular structure compared to small molecule drugs. So if we look at that schematic at the bottom of the slide, they have a linker which covalently attaches the drug to the monoclonal antibody. And maintaining a stable linkage between the antibody and the drug during circulation in the body is really important. So any free drug, particularly any cancer toxic drug, tumor toxic drug that's released prematurely away from the intended site can increase the risk of non-specific toxicity. So the whole point of being able to target the drug is that you can use lower doses of it that will ultimately be delivered to the right location and increase the amount of concentration of that drug at the intended site. So if you're releasing it away from that intended site, there can be, problems of non-specific toxicity. So with that structure, these kind of three-part structure in this sort of simplified schematic, it's really important to understand the properties of each of those components, so the antibody, the linker, and the payload drug, and to understand the characteristics of each step of the production process as well. So that would include the attachment of the drug and the linker, the conjugation of that to the monoclonal antibody, and the subsequent purification steps, as well as the resulting process intermediates. So if you're trying to understand the impurity profile, for example, throughout the different stages of production, you need to be able to accurately and explicitly link analytical data to specific contaminants and degradation products and their structures. But of course, when you're looking at molecules like this, chemical structures aren't really an effective way to represent the monoclonal antibody part of the molecule. They'd be an extraordinarily large structure, for example, and likewise a sequence-based representation that does perhaps fit the monoclonal antibody really well can't really be used to represent the small molecule element. So you need a balance of the two. So in terms of more broader challenges just beyond the sort of structural ones, as you're trying to develop an antibody drug conjugate, the characterization of them is a challenge compared to small molecules. So it's not just trying to understand and characterize those three different elements I mentioned then. It's about understanding the various attributes of that and the ways that you can understand and characterize these molecules is somewhat more complex than a small molecule as well. So, for example, due to the inherent biological variability of producing any kind of biologic, no two batches of a biological therapeutic are actually ever quite the same. And when you think about the regulations for the production that go alongside the production of biological drugs, regulations often require monitoring of both the manufacturing process over time as well as the finished product. So you're monitoring the characteristics of the drug or the, in this case, the ADC over its production process rather than just looking at the final product. So biologics typically include a complex pattern of product and process related impurities, and they're usually heterogeneous mixtures of those impurities, that is, they're usually heterogeneous mixtures of closely related molecular weight and charged isoforms. So it can be really difficult to delineate natural molecular variation in a molecule from a process impurity, especially if those changes are very small. So ultimately, when you're trying to characterize a biologic, and in particular in this case an ADC, it can require quite a large number of different tests, so a wide range of different tests, including both immunochemical and physicochemical property testing. And those kind of come together to confirm not only the identity of the molecule that you're looking at, and also to demonstrate its potency or biological activity, but also to determine its purity, its stability, and ensure that there is consistent quality as well. So why does biologics research and development need better analytical data management solutions? Well, I think I was hinting at it there, that there are a lot of different tests to come together when you're trying to characterize a biologics. And in particular, those large number of different tests typically are conducted on lots of different analytical equipment, so different techniques and different instruments. And as is generally the case in the analytical market, those techniques and instruments lack harmonization of the data formats that they produce, and that historically has led to pretty large issues collating that data. It's not limited to different instrument types, but also more often combining data from the same instrument from different vendors. So, in most companies, that data is also spread across different silos. So not only do you have multiple formats for the same type of test from different instrument vendors, but you may actually store those in different places as well. So those silos might include ELN, LIMS, chromatography data systems, instrument databases, SDMS, or in some cases even things like file shares. And ultimately, you often find that without a robust data management solution for that data, you'll find that people are gluing it all together or trying to connect it together, or at least index where it's stored in something like Excel. So it's acting as a way of navigating through the data rather than actually managing it in an effective and efficient way. And of course, that lack of data management or robust data management system increases the burden on scientists who actually characterize the data. It can really require fairly Herculean effort to bring together all the data from the different instruments, from the different analytics. And you're probably familiar with a quote like this, data scientists spending 80% of their time as data janitors, so doing all the formatting and bringing together of data, and very little of their time actually spent analyzing it. So I know that's specifically referring to data scientists, but I think that data wrangling effort is something that many people in the lab themselves feel today, they're experiencing that problem. Regardless if their job is as a data scientist or a lab scientist, anybody who has to pull data together to do some sort of analysis or characterization has to spend typically far too much time than expected doing that particular part of the job. So, if you borrow from terminology that's used in quality by design or QBD principles, I always think it's really helpful to think of this concept of data within the end with the end in mind. So people talk about design with the end in mind, but if you think about data when you produce it, how it might actually be used down the line. And I think in a similar vein, many of you will have come across the FAIR principles. So ensuring data is findable, accessible, interoperable, and reusable. All that ultimately culminates in that last part, the reusability part of data. So harmonizing and centralizing data from the outset makes the reuse of that data much simpler and ultimately streamlines the characterization of biological drugs. So biologics offer enormous promise, but the complex nature of their development and characterization presents a huge data management challenge for those companies producing them. It's something that needs to be resolved, and we can look at technical solutions that can address that problem for a typical biopharmaceutical organization. And with that, I'd like to hand across to the next presenter and open to questions at the end.
Thanks, Eliot, for that great presentation. I'm sure everyone has a lot of questions, so submit them now, remember, on the panel on your right-hand side, and we will take them all up at the end. Also, a reminder that in the same panel, you can see a handouts section, and there we've just collated some resources for you so that you can learn a bit more about how software can help you with your biologics research. Now, I'll turn it over to Karim.
Thank you for the introduction. Welcome, everyone. We appreciate that you have taken time out of your busy day to attend this webinar. As Eliot stated, there's a large gap in analytical data management requirements for biologics. In fact, we've already been working with some of our customers whose work has shifted towards working with biomolecules. We believe that the Spectris platform, which has been deployed at many organizations worldwide for harmonizing and centralizing analytical data of small molecules, is very well suited to address this need. In this presentation, I'll give you an overview of how the ACDLabs Spectris platform is used for managing analytical data of small molecules. Then I'll show you several recent improvements that have been made to the software specifically for biomolecules. Finally, I'll discuss a use case of how Luminata, an enterprise software based on the Spectris platform, can address challenges of data management and antibody drug conjugate development. Small molecule isolation and characterization requires many different instrument types. There are numerous vendors for each instrument type, and all of them have their own proprietary format and processing software. This leads to many data silos, not to mention a huge burden on training. Unfortunately, because of this, as Eliot stated in his presentation, highly trained scientists spend much of their valuable time as data janitors rather than on doing research. Over the past 2 decades, ACDLabs has worked at building close business relationships with nearly all of the major instrument vendors for each instrument type, and currently our software supports over 150 vendor formats. In the rare situation where we don't have access to the vendor format, we can import their data using one of several generic formats such as ASCII, JCamp, or NetCDF. Once the data is imported, it can be processed in a single software interface with a wide array of technique-specific processing tools. The data can be processed manually or in an automated fashion. Direct integration of chemical structures with the analytical data provides advanced capabilities such as intelligent structure assignment of LC-MS peaks, structure spectrum verification by NMR based on a machine learning or neural net prediction algorithm, and prediction of chromatographic peak retention times based on calculated physiochemical properties of the structure. These tools and many more greatly assist scientists and improve the efficiency of their research. The software also has a flexible template-based reporting system to enable generation of comprehensive reports in many available formats. As you can see from this image, a database record can include a chemical structure, associated spectral data, as well as metadata, which can be configured based on your individual needs. On the right-hand side are instrument parameters, which come right from the raw data from the instrument, and multiple tables which get generated upon data processing. Notice that a single record can have multiple data sets associated with it. This one that we're looking at right now has a carbon-13 NMR spectrum, a 2D NMR data set, as well as a mass spectrum. One key benefit of the Spectris platform is that it works with live analytical data. This means that the data can be reprocessed, reanalyzed, and easily compared with other data sets. This database is also highly searchable. Users can search by structure, substructure, structural similarity. They can search by any of the text fields that are available. And due to the fact that it's live data, you can also search by spectral similarity. We have successfully rolled out several large-scale enterprise deployments of the Spectris platform for various customers around the world. A system can be set up within your organization's own IT infrastructure or within a cloud environment. Different workflows can be set up based on your specific needs. One example can be that analytical data from various instruments can be automatically captured, processed, and stored in a centralized database. The software can also collate associated metadata from different sources, such as an electronic notebook, laboratory information management system, and chemical structures from corporate registry can be attached to the record based on a unique identifier. Users can then access the data with their Spectris client software. If necessary, the data can be further processed or edited and then checked back into the database once it's done. Colleagues with proper access permissions can have read-write or read-only access to the data. Once the data is normalized and standardized in a central database, it can be used to extract additional value. For example, data from a collection of chromatograms or a collection of NMR spectra can be exported from the database in various formats for AI or machine learning systems for the development of new in-house prediction algorithms. If you're interested in finding out more information about this topic, I'd encourage you to look at the available supplementary material on the Pfizer global deployment of their scientific information library based on the Spectris platform. Now I'd like to discuss some recent improvements that have been made to the software specifically for working with biomolecules. This includes new tools for structure representation of peptides and higher order structures such as antibodies. From an analytical data perspective, we have new import capabilities for processed 2D NMR data sets of proteins, improved LC-MS analysis of large molecules, and in silico chromatographic separations of proteins and peptides. In terms of handling biomolecules, we have recently added the Biosequence Tool to our structure drawing package, ChemSketch. This is an interactive and flexible tool to allow users to rapidly draw and visualize peptide sequences. The tool has access to a series of standard amino acids. This is just a short list that's shown here. More are available. It also provides access to non-standard amino acids, as well as end groups and linkers. A peptide can easily be created using the Biosequence Tool. Notice that the formula and molecular weight are automatically calculated. The Biosequence Tool can display the one-letter code as well. Hovering over an amino acid shows a call-out displaying the expanded chemical structure for that portion of the molecule. And a single click can provide the fully expanded chemical structure if desired. We also have categorized templates within our drawing interface. These templates allow you to quickly select standard structure categories, which can be used as is or further modified. If desired, you can create and define your own template category. For example, you can create a category containing structures of your most often used peptides. We have recently added graphic labels to the template window. Here, you can define the image as well as its molecular formula. Incorporating the graphic label to a chemical structure captures the accurate molecular formula and formula weight of the new biomolecule that's generated. Now I'd like to highlight some improvements in analytical data handling. While we have many advanced capabilities for processing NMR data in Spectris, there are some very specialized software packages specifically designed for the analysis and assignment of 2D NMR spectra of proteins. Sarah, which is shown here, is one of those software packages. One of our longstanding customers that has a global data management system for their small molecule research requested to have the ability to import this process data into the Spectris platform. By exporting the data in an intermediate format, we not only were able to import the spectra, but also the protein structure as well as full spectral assignments. Once in the Spectris platform, data could be edited, annotated, reported, and added to the database. As mentioned earlier, MS and LC-MS data in Spectris can be handled in a chemically intelligent manner. The software will guide the assignment of a chemical structure to a peak within an LC-MS dataset. For MS2 data, the software will automatically assign fragment structures to specific ions. This helps for structural characterization. Spectris also has chemometric algorithms that allow users to automatically extract components even when they're buried underneath other peaks. All of these tools allow scientists to work more efficiently. Initially, within Spectris, MS spectra of large molecules were difficult to analyze because they often formed multiply charged species. This produces a complex isotopic cluster, even for a single component as illustrated in the schematic. We have recently incorporated a charge state deconvolution algorithm within the software, which groups together ions from the same component and therefore dramatically simplifies the analysis of these entities. Method development is a keystone activity in drug discovery and development to enable the isolation and identification of entities of interest. Often, temperature and gradient slope are simultaneously optimized so peaks can be separated. For small molecules, the correlation between the retention time of a peak and temperature or gradient are often linear. Therefore, modeling software for chromatograms for these factors are usually very accurate, with correlation between experimental and predicted retention times less than 2%. So as shown in this simplified illustration, as temperature is increased or decreased, the change in retention time of a peak is highly predictable. Not only can modeling result in identifying chromatographic conditions likely to produce good baseline separation between components, but the software can also assist in identifying conditions where the chromatogram is likely to be robust. Looking at this resolution map that plots temperature and gradient, the colors correspond to the resolution between critical pairs of peaks in the chromatogram, where orange is good separation and blue is poor. This area, marked in orange, is robust because any small change in column temperature or gradient will not compromise the quality of the separation. Chromatographic modeling of small molecules is extensively used across the pharmaceutical industry and has resulted in improved quality, better risk management, and considerable time savings. To investigate modeling of proteins, we worked with Patrick Peterson and Mel Ubry, two scientists with decades of experience in chromatographic method development within the pharmaceutical industry. In fact, Patrick is currently at Novo Nordisk, a pharmaceutical company working exclusively with biomolecule therapeutics. In this investigation, what they found was that gradient modeling of peptides and proteins could be modeled accurately with a first-order equation like that of small molecules. Response to temperature changes for proteins and peptides, on the other hand, was nonlinear, as shown in these two charts. Within LC Simulator, our chromatographic modeling software, it's possible to fit chromatographic data with a variety of mathematical models. It was found that a first-order linear model for gradient and a second-order polynomial model for temperature resulted in very good prediction accuracy for both reverse phase and ion exchange chromatography. After this investigation, we have included reverse phase chromatography and ion exchange models for proteins and peptides in our commercial software. So why do we see this behavior for temperature optimization of proteins? One explanation is the following. At higher column temperatures, the peptide or protein is unfolded and the interaction with the stationary phase is similar to what's observed with small molecules. Therefore, you see a close to linear relationship. However, at lower temperatures, intramolecular interactions result in conformational changes of the biomolecule, which leave less interaction with the stationary phase. Therefore, at lower temperatures, these biomolecules behave differently. In a recent study, Ahmad and coworkers from Merck Research Labs conducted an extensive study on modeling a diverse set of proteins ranging from 12 to 670 kilodaltons. For this set of proteins, they confirmed the previous work that was described earlier. That is, it was necessary to use second-degree fit for accurate temperature modeling. They also identified that the addition of a strong cheotropic modifier, such as sodium perchlorate or guanidine hydrochloride, gave very accurate retention predictions with a linear fit for temperature modeling, like what was observed for small molecules. These cheotropic modifiers are likely preventing the intramolecular interactions that were discussed in the previous slide. This finding proves a general approach for the accurate modeling of biomolecules by reverse phase chromatography. Errors between experimental and predicted retention time are similar to that of small molecules, typically less than 2%. In this publication, the group highlighted two use cases. Using a strong cheotropic agent and a linear fit model for gradient and temperature, they were able to successfully predict and achieve baseline separation of a monomer, dimer, trimer, and a high molecular weight species in a vaccine process sample. In the next example, using a secondary polynomial model, they achieved baseline separation of a multi-component monoclonal antibody mixture with less than 0.4% error between experimental and predicted retention times. In the last topic for today, I'll highlight how Luminata can address the challenges of data management in antibody drug conjugate development. Luminata is an enterprise software based on the Spectris platform, which allows multidisciplinary product development teams to answer questions and make decisions with up-to-date information at their fingertips. For greater efficiency, Luminata has also been integrated with other informatics systems such as ELNs, LIMS, and chromatographic data systems. Since Luminata is based on the Spectris platform, it adopts all the data management capabilities that I mentioned earlier. Different project teams can collaborate with real-time access to the software. For example, process chemistry can identify a process scheme and upload reference MS and NMR datasets for the final API. Analytical teams can then analyze the data and identify impurities, marked in orange, for each stage of the process. When a structure is not known, text identifiers can be used. Arrows indicate the fate of each entity from stage to stage. Luminata can also import data from multiple batches and display batch-to-batch variability. This is an interactive system, so if I select an entity, the software displays the analytical data and the associated metadata for that compound. This interface can be configured differently for each group and can handle multiple use cases such as impurity control, force degradation, stability, formulations, as well as supply chain management. Using the new structured representation tools that I highlighted earlier, project teams can detail the process scheme for the development of an antibody drug conjugate. As you can see in this slide, the cytotoxic payload is attached to the peptide linker at stage 2. This product from stage 2 is then coupled with a monoclonal antibody in stage 3. Luminata allows you to carefully document and track impurities or degradants observed at any stage of the process. It also allows you to add live analytical data to any of the entities and compare batch-to-batch variations in order to produce a safe and efficacious biopharmaceutical with live analytical data with quality in mind. If you're interested in finding out more about Luminata for ADC development, please see the white paper that's in the supplementary information for this webinar. Thank you for your attention.